I hope you're all ready to look at life in 3 dimensions. So today, what I'm going to focus on is how looking at things in 3 dimensions suddenly gives you a completely different picture about what you've been thinking about a protein, and how that visualization in 3 dimensions actually is integral to the function of all of these proteins. Today, our learning objectives is going to be looking in 3 dimensions. What can structure tell us? How is structure an integral part of, without structure, why is it integral to understanding functions of all of these things? And how one single mutation in a protein sequence can affect its 3-dimensional structure in a way it changes its behavior completely. And as I told you yesterday, any change in a gene, it gets personal. If it gets personal, that means it affects us, right? So which means that's what we call, if it's not normal, then we call it a disease, right? So that's pretty much what we're going to focus on today, right? So introduction to protein structure, right? So I'm sure all of you have seen all of these pretty pictures, right? And I must tell you that a huge amount of work goes behind before we can actually see this pretty picture, right? That's my expertise. That's what I'm trained in. And so I've gone through the pain and suffering of actually determining the 3-dimensional structures of proteins, right? So what is this? This is the first structure of protein that was solved. And that's something won a Nobel Prize as well. Any guesses of what this structure is? Just fill in the blanks. Anything? The whole, yes, you're right. It's alpha helices. Is that what you're meaning? Or just the blue? Yes, but what is the name of this protein? This is where you kind of guess. It starts, I'm going to give you a clue. It starts with the M. Anything that's actually very important protein in our bodies. Absolutely, Chandrani, and Chandrani is correct. It's actually myoglobin, correct. This was the first structure of a protein that was actually solved. And because it was so important, actually it won a Nobel Prize, right? And then it was shared by Max Perutz and John Kendrew in the 60s, long time ago, in chemistry, and it was divided between these two, right? This was the first time, and you can actually imagine the awe that people had when they actually saw this first structure, right? And those days, we're talking about 50 years ago, how were structures actually solved? They're actually built, literally built, which is called the wire model. And if any of you come to Washington, D.C., and you have an opportunity to go to the National Institutes of Health, they actually have the wire model there. And this is how scientists used to measure angles and all of those things, right? Can you imagine how it was? But it was very precise. And now we don't even know what effort we have to put in to get a structure. Now, this is all the different Nobel Prizes that people have won for structural biology. And you can see pretty much almost every other year, there is a Nobel laureate who has solved the structure of something. So this just tells you how structures are very important, right? Without structures, understanding functions is hard, right? In essence, if you want a Nobel Prize, study structural biology, right? I'm not kidding. Of course, all the easy protein structures have already been solved. So all the ones that are there are all very, very difficult proteins. So any one of you interested in structural biology, I would urge you to pursue structural biology because, we find answers to these things. Of course, part of it is a joke as well, right? So why else would we study a protein structure other than, of course, winning a Nobel Prize, right? Protein structure. So when you look at this structure and when you look at this structure, right, I'm going to take this off here. So when you look at this structure and when you look at this structure, do they look similar to you, to your eyes? The helices are here. Yes, of course. Yes, Betsy, it actually right. This actually looks very similar, right? the structures. But when you align the sequences, they're only 21% identical when you do an alignment of these two protein sequences. Isn't that shocking? Two proteins that have about 20% in sequence similarity or identity still can have the same structure, right? These are different enzymes, right? So what we assume, we make a lot of assumptions in science, right? So what we will say is that in order to understand functions, even when the sequence similarity is very, very low, to find the relatedness between two sequences, and if they have the same function, we just cannot just do a blast and look at them and say, oh my God, they're only 20% in sequence similarity. That means they are not related to each other, which means they probably don't do the same function. And also they don't actually have the same, they are different, but that's not what it means. So that's why in order to understand similarities between two sequences, even if their sequence identities is low, we need to have three-dimensional structures. So you can actually discover relatedness between two proteins that are not similar at sequence, but at the structure level, right? So why study protein structure? Also, although it is a lot of work and a lot of pain and suffering, it helps us to understand function, right? Without protein structures, we can understand functions precisely. Now, you all know this was again Watson and Crick. They actually got the three-dimensional structure. And of course, I have to also mention Rosalind Franklin, right? They actually found out the structure of DNA, three-dimensional structure of DNA. Just imagine if we didn't understand DNA structure today, we won't be doing any of the things we're doing today, because we won't even know what pairs with what, and we wouldn't even know the stacking effects. We wouldn't even know how DNA binds to proteins. We wouldn't even know anything, right? So that was a landmark. And now these are the three people who actually won the Nobel Prize for that, physiology and medicine, right? Now again, why else would we study protein structure? We will study protein structure to understand fundamentals of cell machineries, like protein translation. How are proteins made in the cell? How do proteins actually work? Do you remember any protein that is involved in the whole process of translation? Can you think of any protein that's integral to the whole mechanism of translation? Do you know where proteins are made in the cell? Any guesses? Yes, that's correct. Ribosome, my goodness. Cytoplasm, absolutely. So ribosome, correct? Many of you have said ribosome, absolutely. It's actually where it's one of the most important machinery in the cells. Are there any proteins that are part of the ribosome and the whole translation machinery? That's actually very important. What actually recognizes your mRNA? Of course, there are lots of enzymes. That's correct, Jeffrey. There are lots of enzymes, DNA polymerases and all of those things, right? So this is the three-dimensional structure of ribosome. You know, it's humongous. You know how many years it took them to actually come to this high resolution structure to look at atomic details of every single atom in the whole ribosome machinery? It took them actually 20, 30 years to actually come up with the structure. And the minute we have the structure, we can explain how the tRNA binds, what is the role of the RNA component of these ribosomes, what are all these different proteins actually doing. Exactly, we were able to explain the entire process of translation. And of course, ribosome, the biggest lesson we learned is a ribosome is a ribozyme, which means it's an enzyme, right? So there are 3 tRNA sites. We knew that tRNA played a big role, but we just didn't know where the tRNA is bound and how all of these different sites actually one went to the next and all of those things, right? So based on the three-dimensional structure, we were able to exactly locate where the tRNA binds, what are the different interactions and all of those things. And these are the 3 people who actually won the Nobel Prize. Actually, he passed away unfortunately last year, but they're all there. So then why else would we study protein structure? We study protein structure to understand protein channels, work as they make up 30% of our cells, are membrane proteins and channels. They form all these transporters and all of those things, right? Again, Roderick McKinnon, he actually figured out exactly how things in the cell move in and out of the membrane, right? In and out. Without the three-dimensional atomic resolution and atomic positions of every single atom, we would not know exactly how these channels work and how these actually sit in a membrane, right? This is the potassium channel, and he figured out exactly what every single atom also means. You know, I run an entire course on all of this. So as you know, I'm just giving you just a glimpse of what structural biology can actually help, right? So it's a lot there. So we'll just giving you a little flavor of what structures can help, and you understand. Also, it helps us to understand enzyme mechanisms, how peptidases work. For example, have you all heard of peptidases? Do you all know what peptidases actually do? Just based on this peptide? No? Okay. So does this give you a hint? Cleaves? Yeah, it actually cleaves, but it cleaves specifically. Yes, exactly. Peptide bonds. Correct. Fantastic. You guys are amazing. So this is peptidases, means it cleaves peptide bonds, right? So any enzyme, it actually works only on one or two amino acids, right? It just cleaves something, it breaks down something, that's correct, and all of those things, right? So how did we understand exactly which bond is getting cleaved and how is it getting cleaved, which so that exactly to the details of an atomic, to the atom and to the bond, it actually helps you understand structures. And also structures helps you understand, learn about evolution. You look further back in evolutionary history and figure out how. I actually gave you an example of how two proteins that have very low sequence identities had the similar structures. Here I'm talking to you about two different protein structures, but they actually do the same function, right? So we have conservative evolution, and we have different kinds where actually they come together. Of course, I don't want to confuse you because that's evolution itself is another topic for discussions. We can do it another time, right? So again, it helps us to understand that this binds to the same ligand, this also binds to the same ligand. What is common between these two are they are both dependent on PLP, right? Pyridoxal phosphates, right? And now again, D amino acid aminotransferases and L branch chain amino acid aminotransferases, they also have the same structures, right? So without the structure, we wouldn't actually even know that D amino acid aminotransferase and L branch chain amino acid aminotransferase are actually the same, which means they do the same thing. So again, it helps you understand this, right? Last but not the least, structures help design drugs. Drug design structures are integral to drug design. That is what I do for a living. This is what I do. I look at structures and I do drug designing, drug repurposing. That's actually part of my research. That's what I do for a living. So it's actually phenomenal how to come up. And I'm sure all of you have heard of remdesivir. Does it ring a bell? Have you all heard of remdesivir? It's all over the news these days. Do you, yes? Which context did you hear about remdesivir? COVID, correct? So we heard about COVID, right? So for COVID, exactly. For COVID, they said, oh my goodness, remdesivir actually works, and it actually does not cure them, but at least it minimizes the hospitalization stay for some of the patients. How does remdesivir work? How did they find remdesivir? Through structural biology. They actually docked remdesivir to RNA-dependent RNA polymerase of this COVID. And they said, well, that's how they actually found the remdesivir. So they're actually using it. So there are many drugs that people are desperately trying to find for COVID, including myself. We're doing drug repurposing. How are we doing drug repurposing? We're just using all the drugs that are already out there and trying to dock it to all the different proteins and figuring out the way if it'll actually work and can we explain the mechanism, right? Now, again, these are some more examples. So it's a imatinib. It's a tyrosine kinase inhibitor. Anytime you see IB at the end of any drug, that means it is against a kinase, right? So we have all of this. AB means antibody. So we have all of these nomenclatures. So imatinib, again, was actually the structure is available and it was, imatinib binds to the structure. And this is, again, it's a cholesterol lowering drug, atorvastatin. I'm sure you've all heard of statins. And in fact, statin was one of the things that I put on my slide yesterday when I was talking about SNPs, right? So we have identified some SNPs that actually, some people, why statins work in some people, it does not work in some people. Why does it give adverse effects, side effects in some people? So we actually talked about this yesterday, right? So again, three-dimensional structures, right? This is again, listen, no prill. So this is actually, there are lots. I'm just giving you some examples of how structures were actually used in order to understand the drugs, how the drugs bind and how the drugs interact with the proteins and all of those fun stuff. I'm sure all of you have heard of ibuprofen, correct? This is something that people like me who have headaches all the time from different things actually take a lot, right? Ibuprofen, and it's a popular one, right? So again, it's for pain relieving and inflammation, relieving inflammation. So, this is how it is. So at the end of the day, many proteins are used in treatment of diseases, hormone erythropoietin, and this is, again, it's a hormone, monoclonal antibody. So I said AB, which is an antibody, monoclonal antibody. So this is an antibody molecule. So that actually binds to its, where is the pink that they're talking about? So this is the pink color here they're talking about. So the antibody binds to its antigen binding sites. What are the amino acids that are involved in this binding? All of these things. So, right. So I think I've given you enough examples of how structures are actually used in drug design. And I'm sure you've all heard of this as well, right? ACE, ACE2. I'm sure you've all heard of ACE and ACE2. Again, this has become very popular right now because of COVID again. ACE2, this is not ACE. ACE is downstream of ACE2, which is a receptor that COVID binds to, right? So these are all different examples. You know what? And last but not the least, a picture is worth 1,000 words, right? When you look at the picture, you know exactly where the ligand is binding. This is again an antigen antibody interaction. So exactly, without structures, you won't be able to understand exactly where things bind, what kind of interactions that they actually take, and all of those things, right? Again, so antigen and antibody, the way they work, I'm sure you've all learned, is by shape complementarity, right? And induced fit. So when an antigen binds to an antibody, that is actually true for any ligand protein interaction. They actually, the minute they bind, they induce some conformational changes. And so it's kind of a fit,